Hello, everyone. I'm Malika Ashford. I'm an editor at Genome Web, and I'll be your moderator today. Today's Precision Medicine online webinar is a panel talk, Standardizing Pharmacogenomics Laboratory Processes and Reporting, and it's sponsored by Agena Biosciences. Our speakers today are Kristine Ashcraft, President and founder of YouScript, Victoria Pratt, Director of Scientific Affairs for Pharmacogenetics at Agena Biosciences, and Glenn Sawyer, Agena's Associate Director of Clinical Genetics. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Glenn.
Thank you, Malika. At Agena Bioscience, we're committed to precision medicine. So we're honored to be able to sponsor this webinar with Genome Web to further our knowledge and hear from the thought leaders, some key opinion leaders in pharmacogenomics. So again, thanks to Kristine and Vicky for joining us. Our first question for the panelists: there's a common hurdle to clinical implementation of PGX, and that hurdle is there's a lack of standard terminology regarding metabolizer status. In recent presentations, some of our colleagues at some of the conferences we've been to have shown that various institutions use different wording for specific genotype or star allele characterization within their EHR systems, and this can lead to confusion around patient treatment and dosing. So Vicky, I'll start with you, and I'd like to get your thoughts on standardization of terminology in pharmacogenomics.
So the clinical implementation pharmacogenetic CPIC, clinical implementation, has actually done a harmonization project working with the Dutch Pharmacogenetics group and themselves really trying to standardize that sort of terminology related to metabolizer status, such as for the CYPs, activity scores, also for various genes, and then genes that like SLOCO1B1 that are receptors, having standardized terms around there. So there is a really sort of global or US European effort to sort of standardize that. It still can be a little bit confusing. I know physicians at the university I used to work with, they still got confused. What does intermediate mean? What does poor and normal they understood pretty well, and ultra rapid was probably, but intermediate was still sort of a confusing term for them, even though we have sort of a harmonization around that.
So, and Kristine, in talking with colleagues and your experience in PGX, what have you experienced as far as confusion around terminology and efforts for standardization?
Yeah, I agree with everything that Vicky has stated. I remember, when we started offering pharmacogenetic testing at one of my old employers back in the early 2000s, we called normal metabolizers extensive metabolizers because that's what was in the literature. And we changed it in our reports because I realized it was making doctors very confused. They thought extensive meant more. And so they were wanting to change the way they were prescribing accordingly. And so I think that standardization makes a ton of sense, but we do need to be cognizant of not confusing the providers on the other side. We used to come up for transporters or receptors, hey, this means this, if there wasn't any type of information out there. What we've landed on is if there isn't information on assigning something yet, and those normal metabolizer, poor, intermediate, ultra rapid don't make sense, we just default to variant absent or variant present and keep it at the variant level until a standard comes out, because it helps us avoid confusion in the field.
And do either of you have examples, Vicky? I know you touched on some where you've actually seen this. You've seen confusion from the clinicians, and what can that lead to? What do you think are the downstream ramifications of that?
So many drugs don't really have information, like if you're an intermediate metabolizer, how in the drug label, in the FDA drug labeling, about how to dose those patients. So that does cause some confusion by practicing physicians. So many times, many laboratories will point physicians to resources in CPIC or PharmGKB where they can put the metabolizer status in, or they can put the genotype in, and help them make dosing decisions for their patients. Another gene where I think there's been a lot of confusion in the nomenclature is for DPYD. So in the drug labeling in Europe, the EMA has star alleles, and in US and in PharmVar, which is a resource that catalogs all the sort of alleles or variants in many of the pharmacogenes. We have star alleles in the EMEA label in Europe, but in the US we have RSIDs or HGVS, Human Genome Variant Society language. So one variant can have 3 or 4 different names, and that's not alone confusing for physicians, can also be confusing for patients too.
And Kristine, to follow up on that, because obviously with your expertise with reporting with YouScript, how would you suggest, or any thoughts on how to handle some of that information, like Vicky's mentioned, like around DPYD?
Right. So we align with any CPIC guidelines that come out, and they put out some great guidelines around DPYD specifically. But I think when we are able, we try to make sure it is known all of the RSIDs that we're looking at in order to land on something. It seems to be the most understandable out there, but we also have other standards stored in the background, because if you're integrating this type of information into different electronic health record systems, different pharmacy management systems, they haven't even aligned on drug identifiers across healthcare IT. So you do have to map things in the back end, but I think that's the simplest and easiest to understand.
Thank you both. Vicky, any additional thoughts here, Vicky, before we move on?
Nope.
Thank you both. Appreciate that. It's, as always, great insight. And Kristine, we'll start with you on this question. This one is, the specific content that's offered within a PGX test, that also lacks standardization, right? So some labs offer one set of genes and alleles, while another lab may offer a different set. So in your opinion, what's the best approach to standardization of PGX testing content and the test ordering?
Right. So, I mean, we know in an ideal world, every panel would cover all the genes with CPIC level A or B guidance or FDA actionable or higher, but that's just not feasible from a cost and turnaround perspective. So you do have to make choices. I do think if AMP guidance exists, as it does for more and more genes that are relevant in pharmacogenomics, Tier 1 coverage should be absolutely required for reimbursement. So if there are any payers on this call, update your policy. Don't cover it if it doesn't have Tier 1 coverage. That's my recommendation. In my opinion, it is okay to include developing evidence genes, but it is not okay to recommend drug or dose changes if the evidence is not there yet. It is okay to give a summary of where the evidence is at to a healthcare provider. So if they want to use their own clinical judgment to decide to do something with that, fine. But we as laboratories should not be telling people to change drugs or doses if the evidence doesn't align with it yet. I don't personally, though, see a problem with including developing evidence genes. Again, there's a lot of back and forth. When you're talking to the healthcare providers ordering this test, some of them really want what we PG experts would call a questionable gene on a panel. But I don't think there's an issue with it, again, as long as you're not telling people to act on it, such as SLC6A4.
Well, now I have a new name for my next band, the PG Experts. So I appreciate that insight. That's awesome. Appreciate it. And Vicky, with your expertise, in writing a lot of the guidelines as well, anything to add here? Your thoughts on, what content should be added and how, what tests should be ordering as far as standardization?
Well, thank you, Kristine, for introducing my passion project, the AMP recommendations, and totally agree with Kristine that every tier 1 variant really should be in clinical testing. We want to make sure when we're writing the AMP guidelines or recommendations that it's really a pan-ethnic testing, that we're appropriately testing all ethnic backgrounds, where we have concern in some tests that don't cover that sort of pan-ethnic background or genetic ancestry that we have a lot of concerns about. I guess I'm going to slightly disagree with Kristine about having lower evidence genes on there. I'm okay that it's on there for research, as long as it's really annotated very well that the evidence is not good around this. There shouldn't be any drug dosing guidelines. But to me, that's more on a research basis. And I don't know if, maybe we should be marking these that this is, whether we call it level, we use the CPIC guidance, or even if there's not even CPIC evidence levels around that will really mark that this is not ready for prime time. It's not ready for dosing. There are no dosing guidelines. I just get really concerned about some of that lower evidence genes, and I get doing research, and did a lot of that when I was at the university, but we had it under an IRB and informed consent information for our patients and physicians.
Well, I'll just chime in there for a point. So recently a publication came out in the Journal of Clinical Oncology that the American Cancer Society Cancer Action Network helped publish around making sure there was equity and inclusion in pharmacogenomics and oncology. And one of the things that was interesting is how many patients you would need to have in a clinical study for oncology to find these variants that don't occur in the general population at a very high rate, but maybe do in a specific population with certain ancestry in common. And I don't think we're going to be able to find certain patterns, especially for underrepresented ancestries, without including these research genes in a growing repository of real-world evidence. So like YouScript has been saving every drug-gene interaction from our integrations in EHRs for a very long time. We haven't done anything with it yet, but I think we're going to get to a point where, especially if we drive for this to be stored discreetly in every EHR, or in some central repository so it can keep being used, that we can move things from developing evidence to actionable. And the only way to do that is to have that kind of information at a broader scale than typical clinical studies have allowed for.
That's a good point, Kristine. Thank you. And that leads to me, follow up to that is me thinking, because we do need, to keep gathering that information on genes we may not have enough information on now, or continue to have that repository. Any thoughts here on, as far as the standardization for like reporting, like what gets reported out? I know you talked about, the level of evidence and the tiers. Any thoughts there where you still can, if you have a test that does run all of those genes and those targets, what gets masked, how you report out? Any thoughts there on how that should be approached?
So reporting, I think that you need to kind of keep at the top of mind elevating actionability. Most providers have just several minutes with a patient, and we need to elevate the things that are what I call EADGIs, evidence-based actionable drug-gene interactions. So what do I need to act upon? Okay, I have a 2C19 for metabolizer and clopidogrel. let us get them off that. That is something that needs to be front and center on the report. Okay, maybe I have some information that says you should consider a dose change for something else. Make sure that is front and center on the report. And make sure it's not just in one clinical area. We actually collaborated with Christ Hospital Health Network on an assessment of 30,000 patients that had had panel testing. And what we found that almost 60% had a PGX interaction in more than one clinical area. That means if you only focus on the behavioral health meds or only focus on the cardiology meds, you are missing information that can impact the safety and efficacy of medications. And for me, that is not okay. Say the second thing in reporting that we really need to think about is empowering the patient. We have not fixed the portability issue for pharmacogenetic test results yet. I would encourage any lab to make sure you make results available through FHIR API. There are FHIR standards for pharmacogenomics. Make sure you can at least do that. But most of the time, those results get lost in a PDF in the EHR of the ordering provider. What we need to do is make that information portable for the patient, at least to some degree. So, hey, patient, this is why this information is important. Here is how you share it with all of your other healthcare providers, and you should do that for life. This is something that can, impact lots and lots of medications. In the recent European study, they gave the patients a QR code that was always having updated information. Great. Historically for YouScript, we gave people little patient cards so that any of their healthcare providers, if they were willing to go to YouScript.net and create a free 30-day trial, they could pull the patient's genetic test results in and audition a drug gene pair. But those are key things on the reports, aside from aligning again with CPIC guidance, making sure it's clear what you're testing, and beyond the detail level of star 3, you need to say what's going into that star 3. I think those are the things that I want to keep top of mind.
So I want to, I do love the idea, and I know some of those QR codes have worked. We piloted that when I was at the university, and many of the patients, we did some follow up with the patients. Many of them didn't remember that they got the QR code, what that gene card meant, what they were supposed to do with it. They sort of forgot about that. Obviously there's very empowered patients and very proactive patients that have that information, because I've had patients come up to me and say, by the way, I'm a blah, blah, blah in this gene, and tell me a lot of information about it. But then on the other hand, we have other patients that don't. So I do think it's important to empower the patient. I don't know the best method around that. To make sure it is democratized to all patients, whether they have high health literacy or lower health literacy, how to ensure those patients with lower health literacy, how to empower them.
Well, I think one way, although I know you think it's unlikely to move forward in our current Congress, but we are getting the Right Drug Dose Now Act reintroduced by Swalwell and Crenshaw this year. This is an act that would help address that by making sure that we don't just flag for drug-drug interactions, but also drug-gene interactions in the electronic health record and pharmacy software systems. And that's key. I think it takes it off the patient, but we need to be doing it somewhere, and we're not, making that portable and long-lived.
Kristine and I, we talk a lot at the same conferences, and we talk all the time. And I tell her, nothing's moving through Congress. Well, they can barely fund the government, for another week or two. So,
well, I will say we're covering pharmacogenetic testing for Medicare patients if it aligns with CPIC A or B or FDA guidance, and have, for years, right, since 2020. We are spending so much money on adverse drug events, especially in our seniors. And it is only getting worse with the silver tsunami with these aging baby boomers. I think it's like 17,000 a day aging into Medicare. We could save a significant amount of healthcare dollars if we did a better job managing adverse drug events, reducing that. And PGX is a piece of that. You know, that European study, 30% reduction in just 12 weeks with PGX panels. You take a $528 billion national spend on adverse drug events in the US, and that's dated, that's 2016. That is a big ability to save a lot of money and lives.
Well, and there was the Kentucky Teachers Union study that was in the Medicare population that did show significant health economic savings. We're derailing Glenn as usual.
No, this is great. I'm just sitting because this is this is exactly why we love to have these panel talks. Because we love hearing it because we know, and that's why we have just a question to prompt, right? Because we know that these are going to spur great discussion, which leads to the third question here that I'll move to. And Vicky, we'll start with you on this one. Because in addition to the test content itself, there are differences from lab to lab in their approaches to PGX implementation. So what do you feel standardization in the laboratory should consist of in terms of verification, validation, and interpretation?
So another one of my other passion projects is working with the CDC genetic testing reference material group, or the GetRM, to ensure that there is reference materials or positive controls for laboratory for their validation and verification of whatever test or platform that they're using. And it's a quality control to ensure that there is that the test accurately can test for specific variants, star alleles in the pharmacogenes. We've done and it started off when I was trying to validate a test in my laboratory, and I didn't have positive controls for many of the variants. They just didn't exist. And so started working with the GetRM, and we've characterized more than 300 samples now that for various variants in many of the pharmacogenes. So there is some sort of reference materials to start with that validation and verification. And there's guidelines from the College of American Pathologists that suggest that there has to be a minimum of 20 samples. The American College of Medical Genetics and Genomics recommendations for pharmacogenetics actually recommended that if you're especially doing CYP2D6, because there's so much variability in that gene with copy number and hybrids and single variants, that really 20 is not adequate for just that gene, that you need actually a lot more. Maybe you need maybe 50 might be appropriate for that gene. There are guidances out there for validation and verification, and ensuring that even the CLIA, that you have to have analytical sensitivity, analytical specificity, your accuracy studies, your precision studies, robustness studies, all of that goes into a typical analytical validation.
Thank you, Vicky. And Kristine, any thoughts on, like, from the interpretation side and again on the reporting aspect, any gotchas or watch outs when people are starting up and trying to get their panels off the ground, and they may approach you saying, hey, what's the best way to interpret this data?
Yeah, I mean, I think making sure that you're adhering to the guidelines. I would highly recommend that any laboratory be part of the CAP PGX proficiency program, so that you can make sure that you're aligning. And I think, again, we should make AMP Tier 1 a minimum for that, because even if you're calling right for what you're doing at your laboratory, I don't think it should necessarily meet that bar if there isn't AMP Tier 1 coverage at a minimum.
Yeah.
And I will also say so CAP covers a specific number of genes. They have an alternative assessment program for other genes that are less commonly performed. So it requires at least 3 laboratories to be performing that testing. I worked with CAP to set that up. Since the GetRM project has a lot of information on a lot of other genes, they also have some information on those more or less commonly tested genes that and that program, that alternative assessment program, is also key, that is run through CAP.
And Vicky, one of the things that we often hear from like new labs that are starting out or labs that are looking to get into PGX, one of the hurdles, challenges to getting up quickly is actually procurement of samples, right? So you've already mentioned GetRM, and then we've talked about the organizations through CAP. Any suggestions on labs that are any organizations or contacts they should reach out to that are looking to start up and actually get samples other than GetRM, or you feel those are the best sources?
I do feel Coriell and GetRM are the best sources. There are a few commercial vendors that sell controls or some sort of control material for certain genes. It's not all of the genes. Some of the sourcing is actually clinical samples, whether it be blood samples or buccal swab or saliva that have been tested in another laboratory. If the laboratory belongs to AMP, the Association for Molecular Pathology, often they can actually just go on CHAMP and ask to exchange samples with other laboratories, and many in the CHAMP community will help laboratories get set up and obtain samples that maybe have been previously tested and send them to laboratories that are starting up.
Yeah. And I will say in those labs that are in states that require a Medicare Z code and technical assessment, just validating with Coriell or GetRM samples was not enough. You did have to actually validate on a secondary methodology, so either trading samples with another lab or running it on a different platform than what you generally run your samples on in your own laboratory if you're capable of that. It was a requirement as part of the validation they were looking for.
And that brings up a great point, Kristine. And I'm going to veer off the standard script here because you brought up a good point with Z codes. Any thoughts on challenges or what you've seen, how you've seen laboratories be successful procuring Z codes and being able to implement those?
Great. Well, I think that they have changed the the questions and what they were recommending people submit over time to try to make it a little more clear. But in terms of validation, that really makes sense. That's a little bit out of my wheelhouse. I would lean on Vicky for that, but I would say it's a lot better to validate. The other thing they do require is that you need to include the information that the doctor has to act on for the drugs that inspired that order. So you've got Medicare LCDs that are like, hey, we'll cover this if you're already prescribing a med and it's causing problems or not working, or you're considering prescribing a med that has this high level of evidence, CPIC A or B, FDA actionable or higher. But on the report, you need to tell people how to act on that, or the healthcare providers, which is really kind of a little bit opposed in some ways to some other information we're getting out there. But I would encourage people to make sure they're capturing that information and providing, again, that elevated actionability. I always have said to YouScript employees historically, what would you want a healthcare provider to see if they were treating your loved one, your mom, your kid? Right? Put that front and center.
Well, and I'm going to sort of go back to what you were asking, Glenn, about Z code descriptors and the MolDX program. We are seeing some commercial providers now, commercial insurers, payers now requiring Z codes for laboratories. So it wasn't just the Medicare population; it's now commercial payers. And so I think laboratories that are starting up and thinking about this really need to be aware. And I think actually the deadline for one large commercial payer is coming up in April. And if they're getting significant reimbursement from that commercial payer, they need to be aware of that.
Yeah, thank you both. I appreciate it. That's great insight, because I know that was not right on script with what we had talked about, but I think it's an important topic that the audience would like to hear. So thank you both for your insights there. The next question, we'll start with you, Kristine. What are some of the critical success factors laboratories should consider when developing their PGX reporting capabilities? I know we've already touched on this slightly before when we were talking about content, but any additional thoughts here on critical success factors?
I mean, the critical success factors, I already referred to a few of them, but just to recap, I would say you want to make sure you're aligning with standards that have been put out there. I mean, CPIC has been around since 2009. They are the guidelines body. Make sure your reports align with anything that they have out there. And the most recent guidance, I've seen some that do refer to it, but it's an outdated guidance. It has an updated guidance and people haven't realigned. And that can take time. It's not like laboratories can turn around and suddenly update things the next day. But make sure you have a process in place to understand those changes are coming. And that you have the capabilities in your lab to make those updates when they become necessary. The elevation of the actionability, I think, is very much key. You can't just generate a report that says, hey, this patient is star 1, star 3 intermediate metabolizer, have a nice day. Again, what would you want them to see if that was somebody that you loved that was on the other end of that report? You want to make sure they're getting the information they need to both help that patient now and going forward as much as possible. I'm not a huge fan of static reports, but they're all we have sometimes, but you should at least create a way for somebody to get more up-to-date information periodically.
Thank you, Kristine. Vicky, any thoughts here as well for critical success factors for reporting in addition to what Kristine has mentioned?
And Kristine touched on it, that laboratory reports are static. They're based on the information that's available at the time. In my laboratory experience, at one point prior to the CPIC harmonization project, a star 1, star 4 was in some called a normal metabolizer, and by others an intermediate metabolizer. And some of this nomenclature has evolved over time. And so the lab reported what was known at that time. At my institution, we instituted clinical decision support. So even though that report was static and the genotype was static, but the information was not static. So the clinical decision support was updated and evolved and continues to evolve. But sometimes it may not evolve as quickly as the information that may be on the CPIC website or FDA website or various other things, just because pharmacogenetics is a quickly evolving area, and trying to keep up with that, it is an expense on the healthcare systems to keep updating the CDS as our knowledge improves over time.
I will chime in here. I will say I've seen a lot of it's not good enough unless we invent it here, especially in academic medical centers around clinical decision support. YouScript has existed in some form since the early 2000s. We've been integrating an electronic health record since 2013. We run on CDS hook APIs, so we can actually create a BPA based on an evidence threshold. So if CPIC level A or B says I should change a drug or a dose, and there is a PGX interaction, interrupt that prescription when the medication is selected and pop that up right then and there. So rather than having to do a drug gene pair at a time, you can actually automate things to say, hey, if there's a major interaction involving a flag to change a drug, I want to flag to order that test, or you kind of setting rules engines instead of doing things binary pair at a time. The other thing that I find a little concerning, maybe a lot, although we're still aligning, is the lack of integrating phenoconversion quite often. This is especially common in patients taking multiple medications. So ideally, reports don't just look at drug gene, but also drug. And if that needs a pharmacist, as KTRS did, they had a pharmacist not just say, hey, here are the gene interactions that need to be resolved, but here are the drug-drug interactions and the drug-drug gene interactions. You need to look at this holistically. And we're not doing ourselves a favor by just flagging, for binary alert after binary alert.
And I totally agree. Phenoconversion is a really huge area right now with the drug-drug interaction, the drug-drug gene interactions that really are adding to what's going on with patients.
And thank you both, because it's good insight. And you mentioned it because we're talking about not just, not looking at just single pairs at a time. And Kristine, earlier you talked about different areas, right? Like whether it be cardio or psychotropic or pain, right? Not looking at those discretely. So thoughts there as well. And when deciding on how to develop the reporting capabilities.
So you definitely need to report across all clinical areas that are impacted, certainly for the medications that are being prescribed, but that's really the importance of pushing for integrated clinical decision support. It's again why I think the Right Drug Dose Now Act is so important. It also calls for educating all sorts of healthcare providers on how pharmacogenomics can help reduce our adverse drug event epidemic. But until we make it a priority to make it easy for providers to know when to order testing or make preemptive testing across the board of thing, and how to act on that in their busy clinical workflow. So you shouldn't be popping up something that just says, hey, stop taking Plavix or clopidogrel, Effient or Brilinta will work if Brilinta is going to cause an interaction with another drug on their regimen. Tell them something personalized right in their clinical workflow that makes it easy. We have the technology to do that, but we have not changed the way that we prescribe medications for decades for the most part, except for in these pockets where PGX is starting to be integrated.
Again, great insight. Appreciate that. Thanks for that perspective. Vicky, we'll start with you on this next question that we have. So in terms of reporting, what aspects do you feel need to be standardized to accelerate the adoption and implementation of PGX in the clinical setting? I know throughout the course of the discussion, we've already been touching on these, but any thoughts or any additional things to consider here?
Well, I do think we've touched on that. I really think having high-level evidence, whether FDA evidence, CPIC level AB, to have that in the tests that are being performed. I do think it's confusing, and I'll go back that we've talked about a little bit, having lower evidence. I always get concerned about how, even though we can flag it and say that there's a lower level evidence, how is the patient perceiving that? How is the physician perceiving that evidence, and are they making actions on that, and how that goes? So really keeping that at that sort of high level of evidence, I think, is important.
Kristine, thoughts here as well? Any addition? Again, I know we've touched on this throughout the course of the questions, but anything else come to mind here?
I mean, as I said earlier, the AMP Tier 1 at a minimum. I know that Access DX, our associated lab with YouScript, also has all the AMP Tier 2. And certainly include them if you're capable, right? We want to continue expanding that information. The CAP program, participating in that. There are so many things that we can do to make sure it doesn't behoove anybody in the PGX industry to not make sure that they're doing the best job they can. to deliver information that can help optimize, the safety and efficacy of medications. We're all out there doing this because we know it's the right thing for patient care, and we need to be just as committed to make sure, we're doing the absolute best job we can.
Yeah, and the passion is evident. So that's what I appreciate you both for sharing those insights and your thoughts. The last question that I have for both of you is, we've obviously circled around a lot of different topics and touched on a lot here for pharmacogenomics, but are there any other considerations or things related to PGX standardization that keep you up at night, or things that we haven't already discussed to this point? And Vicky, we'll start with you.
There's a couple things that keep me up at night. Some of the things that I worry about is tests out there or labs that aren't testing appropriately a sort of pan-ethnic population, that I'm going to sort of just say they're only testing white variants. In the UPGX program that Kristine mentioned in Europe, that was our research project that was done in Europe, was largely only testing white people variants, and they do acknowledge in their paper that they didn't include variants from other genetic or some other genetic ancestries. So that sort of just, I think we do a disservice. We've seen old clinical trials with warfarin where the study only included the white people variants again, and it didn't, and these trials didn't work in people of other genetic ancestries. So that's one of the things that really worries me. Again, that's why part of my passion project with our AMP recommendations is really making sure it's pan-ethnic, but we're only using the information that we have curated at the time through CPIC and PharmGKB. So there may be other ethnicities out there that inadvertently get overlooked because we don't know enough. Maybe some of the South American variants, we just don't know enough about those or have enough information to include those when we're making our recommendations. So those are that's where, I think we're more of a global community. I mean, there are pockets that are less global than others, but we're a global community, and ensuring that there's appropriate access for everybody, or appropriate testing, I should say, for everybody that gets PGX.
It's a great point. I'm very concerned about equity and inclusion. And, there are still, back to the AMP Tier 1, some major labs that are missing variants, for example, that are present in up to 10% of patients of African ancestry that are loss of function variants. Like that, for major SIPs, like that's not okay. And certainly the healthcare providers on the other end, they're not us. They don't know that when they look at a test. They don't know that for the patient that they're treating right then that happens to have African ancestry, it could, be inaccurate a good percentage of the time. And that's something we need to do a lot better with. I think the other thing that kind of keeps me up at night, I am a huge, huge fan of standardization. And I want us to get to a place where we all agree. I think most of the time when we're at least 80% in agreement, most of the time when we talk. And I think that, that should be enough. We shouldn't let perfection be the enemy of good. If you look at drug-drug interaction content, it doesn't align. From the different drug-drug interaction content providers in a lot of different areas. There's a number of publications here showing that, they don't align maybe 20% of the time. And that's for drug-drug interaction content that's been around for a very long time. We're never going to align 100%, but there are a lot of things we can agree on. We need to agree on them, and we need to really push forward how to get this integrated, not just at the academic medical centers, but in the community health systems, in the rural health systems, in the clinics. You know, right now, this is way too segregated as a tool that can really help us optimize medication management. We're losing a life every 2 minutes in this country to non-optimized medications, and we know pharmacogenomics can help reduce that toll by like 30% or more. So I encourage everybody to align with standards, but I also encourage us to make sure we're pushing for this to become standard of care as it should be, and not wait until everything is perfectly aligned and out there, because we can already do a lot of good.
I love that perspective. I love that thought. And along those lines, as far as the pushing, right? Because one of the things that we often hear for the challenges for adoption is, well, where's the evidence with the health economics, right? And so both of you have already mentioned some of those, like where we talked the Kentucky Teachers Union, right? And then some others you mentioned the European study. So any thoughts there for our audience that may be trying to do that pushing, but they're looking to have that supporting evidence to be able to say, hey, here, look, here's some evidence, right? Any thoughts on that evidence that they could provide?
Yeah, I have thoughts, unless Vicky wants.
I'll let you jump. I mean, there's, you know, I'm like going, I don't even know where, there's like so many places to start, but you go ahead.
So I actually co-authored studies a number of years ago. The first one we did in patients 65 and older taking at least 3 medications, one of which was impacted by PGX. We provided a PGX report that highlighted any drug or gene interactions where there was drug or dose changes needed. The providers acted on them about half the time, and we saw I believe 71% reduction in emergency room visits, 39% reduction in hospitalizations in just 4 months of follow-up, saving an estimated $11.32 per patient in $2015 in just 4 months in Medicare patients. That had a 4-to-1 matched historic control, so we got this pushback. Oh, not the gold standard. So we did a small prospective randomized controlled trial. This was done in patients that were flagged as being at high risk of readmission in a program that was already doing an exceptional job. They were in the top 10th percentile for lowest readmission rates in the country. And that's because they actually had pharmacists visit patients in the home, because medication-related issues were one of the main reasons patients ended up back in the hospital, right? So pharmacists visiting these patients in the home, they either randomized patients with PGX-impacted medications to treatment as usual, high-touch pharmacy care, or those pharmacists were given access to pharmacogenetic panel testing and clinical decision support tools that incorporate, well, YouScript, PGX, and drug-drug interactions. And what they saw in just 60 days, 52% reduction in readmissions, 42% reduction in emergency room visits, not an endpoint of the study, but 85% fewer deaths, saved over 4,300 patients in 60 days. A meta-analysis came out a few years ago that looked at a ton of different studies. Did this impact clinical management? Did this reduce hospitalizations? Those studies I just cited were in that meta-analysis, but as you know, you're going up, right? Hey, this is a prospective randomized controlled trial. This is now a meta-analysis. There was a significant impact on medication changes in the PGX arm and a dramatic reduction in hospitalizations across multiple studies. We all know those are very big healthcare resource utilization or healthcare cost drivers. The data is out there. We just need to do a better job of getting it in front of people, I think.
Thank you. I love that perspective.
And getting it to getting it to the payers. There's definitely evidence review bodies such as the Hayes Group and others that don't necessarily have that PGX expertise, but and I know many payers rely on some of those review bodies, and you know there are still concerns that NCCN doesn't have DPYD testing in its recommendations. There was a citizen petition to try to push some of that information, and payers do look at some of these guideline bodies like NCCN related to that. So there's still lots of areas or room for improvement of getting that evidence, getting that right evidence there, getting that information to the right bodies.
I have a major hypothesis that science is always better than guessing. So far, I'm batting 100% in studies to validate that, strangely.
That's awesome. Well, I can't thank you enough, because the passion is evident in both of you, right? And we know that this group and the PGX community and the colleagues that we interact with, that are so passionate about PGX because they know the good that this can do for patients. I can't thank you enough for the advocates that you are and your expertise. And again, thank you for the panel discussion. And with that, I know in the interest, it's been a great discussion. We've had a lot of questions come up through the Q&A, but before that, I don't want to skip the fact that, Kristine, wasn't there an important date coming up that you wanted to mention to the audience?
Yes, thank you. So March 24th is National Adverse Drug Event Awareness Day. I've thrown out some statistics earlier, but back in 1998, there was a cited study showing we were losing 106,000 American lives a year to adverse drug events. Another publication came out in 2016 showing that number was now at 275,000. If that trend continues upward, we're losing about 350,000 American lives to adverse drug events this year. That is 5 full 737s falling from the sky every day with all lives on board lost. If that were actually happening in that viewable of a way, there would be a national priority to address that as soon as possible. So if you go to fourthcause.org, you can learn more about efforts to both spread the word about this and also how PGX can help improve that. But thank you.
No, thank you, Kristine. I appreciate that. And I'll hand it back over to Malika from Genome Web to help us with the Q&A.
Yeah, thank you so much, everyone. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. So let's see if we can get through some of these questions. I think there were a number of questions kind of about the practicalities of, successful laboratory standardization, including, how many samples, you should be ideally using for validation. In terms of test design, are there specific platforms that are endorsed, or are recognized as better than others? Any technologies that are not recommended? Any thoughts on the utility of sequencing? Is that something that, might help in the future to, future-proof for the evolution of tests over time? Can you guys speak to some of those questions?
I think I can jump in on some of that. Agena is sponsoring this. Obviously, Agena is a good platform. There are many other wonderful platforms out there. And I would just be cognizant of when you're thinking about an exome platform, depending on some of the my former group, we've done some validations on it. There are many of the PGX genes have deep intronic variants that aren't covered well by exome or exome platforms. That's why some of the targeted works pretty well. Also with some of the exome platforms, it can't recognize copy number, issues around copy number very well in some of that data. So that can be problematic. I think genomes are going to be better if you're looking at a sequencing or a sequencing-based platform. Genomes are going to be better. You're also going to see a lot of variants of unknown significance and trying to figure that out. One of the GetRM studies that we did, we looked at sequencing in the 2C9, 2C19, 2C8 genes, and we saw lots of variants of unknown significance, and trying to figure them out it can be challenging for labs that incorporate that. I do think there's going to be guidelines soon, or recommendations soon from groups that are looking at that right now.
Okay, thank you. In terms of targets, to include based on society guidelines, is it reasonable to think that, there's ever going to be complete agreement on, a guideline to use, will there ever be one thing to rule them all?
No, nobody agrees on anything.
Yeah.
Did Kristine just say nobody agrees
I think we agree on 80%. I said
We agree on 80%.
Yeah.
And the AMP tier 1 recommendations. There we go. AMP tier 1 recommendations we agree on. But labs are always, my test is going to be better because I have the AMP tier 1, tier 2, and plus other variants that may have no unknown value in them, or I've got genes of unknown value in there. But yeah, so 80% we'll agree on.
What about, the process of reviewing and updating tests as, new information comes to light? How often, should a lab be, looking at the content of the report and reevaluating that? And how do you deal with a future discovery, that wasn't included in a patient's initial or historic test report in terms of recontact or things like that?
So there are guidelines and recommendations around some of that from the rare disease testing world by the American College of Medical Genetics, especially if you see some sort of drug interactions that's not explained by their pharmacogenetic results. I know when I was at the academic medical center, we probably updated our panel of either taking genes out or variants out, and adding variants and genes as that knowledge evolved. We probably did it at a minimum at least once or twice a year. And so somebody who got the test, our PGX test when we first started the lab, was, while much of the content was there, more than 80% of the content didn't change. There was probably some of that 20% that did change and evolved over time as knowledge evolved.
Yeah. I would just say future proofing. I mentioned it before, but to Vicky's point, you should be set up to make updates at least once per year if you are doing pharmacogenomic testing.
And I'll chime in as well, because from a vendor standpoint, it's a great question. It's a fair question, right? If we're providing panels, how often are we updating and thinking about them? That's why we have, and we consult the expertise of like Kristine and like Vicky, right? And what we feel we should have in there in a minimum, but you also have to have the flexibility as a vendor to be able to add content as needed, right? And so you want to find someone that's able to do that for you. Not only the content that's already recommended and what you feel should be standardized, but also the flexibility to add additional content as more evidence becomes known.
Great, thank you. I don't want to get into the question of reimbursement in any detail, but we did have a couple of questions. And you guys mentioned that this has been a historic challenge. You mentioned some of the added requirements, for Medicare in certain states. But I've been writing about pharmacogenomics for more than a decade, and I feel like every year it's like, this is the year. So is this the year in terms of a prime time utilization and more widespread reimbursement? Are we at the point where we can start making that case?
Well, the Medicare LCD started rolling out in 2020 that aligned with CPIC, A or B or FDA guidance.
And did include AMP, tier 1.
Awesome. And then the American Cancer Society Cancer Action Network has been working on biomarker legislation. Last year, NCOIL, the National Coalition of Insurance Legislators, actually endorsed this legislation as model legislation that all states should adopt. Typically, when that happens, it rolls across the country in a few years. And that legislation includes coverage for pharmacogenomics as well. However, it doesn't mean the insurers, commercial and Medicaid, in those states that it's going live or going to go live in the future, are going to instantly start covering this. So you do have to be set up not only to do a great test, but also to make sure you're submitting the correct information. These are the drugs that triggered it. These are the genes. but also having a ready to go appeals letter or even sending it in that says, hey, you need to cover this because of XYZ legislation or it aligns with the Medicare LCD. We are going to have to fight a little bit, but I think everybody knows kind of the key stoppers, right? Medicare reimbursement, integrated clinical decision support. Those are starting to go away.
Great. So we've run out of time for today, but I'd like to thank Kristine Ashcraft, Victoria Pratt, and Glenn Sawyer, of course, and our sponsor, Agena. If we didn't have time to get to your question, we will do our best to follow up with our experts. And as a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thanks so much for joining us.
Thank you.